Good morning, everybody. My name is Aaron Del Duca. I'm the VP Technology at DNA Genotek, and I lead the microbiome program here. I'm very happy to welcome everyone to standardization and microbiome measurements. We'll be talking about a range of topics from sampling through to in vitro wet lab methodologies and in silico analysis strategies. We're going to let the participants have a few more minutes to register, sign in, and get set up. Very pleased that we have a record-breaking list of attendees. I'd like to thank the Microbiome Times for helping us coordinate this. Thanks, Nick and team. And with us today, we have a few presenters. It's an international group. Here in Canada, where DNA Genotek is headquartered, we have Evgeny Dukhanin, who's a senior scientist here in our R&D team. Evgeny is a molecular biologist by training, and he specializes in microbiome research and development. He focuses on formulations and devices that facilitate stabilization of a wide variety of biological material. At DNA Genotek, he leads a team of scientists focused on microbiome and genomic sampling and stabilization technologies, and has developed quite a bit of our intellectual property portfolio here, and is a wizard in terms of making sure that these devices integrate into the most commonly used methodologies out there in the field. With us from the Twin Cities is Daryl Gohl, Chief Scientific Officer at CoreBiome. Daryl is a molecular biologist and geneticist with a really strong track record of developing novel tools for answering really important biological questions. He's been granted a few patents as well, and his know-how has really built the foundation for CoreBiome's technology platforms. He directs R&D at CoreBiome, and he studied at Northwestern, Princeton, and Stanford, and brings a lot of that know-how to bear on our customer services projects through the CoreBiome team. And lastly, we have Dan Knights also joining us from the Twin Cities. Dan is the CEO at CoreBiome and is a computer scientist and computational biologist by training, and a globally recognized expert in microbiome informatics, who's had a hand at developing some of the leading methodologies for analyzing microbiome big data. He directs informatics and software development at CoreBiome, and has an educational track record beginning at Middlebury, heading over to the University of Colorado with Rob Knight while he was still there, and then over to Harvard Medical School with the Slouches on the East Coast, pioneering some microbiome analysis methodology there. All right, so I think the attendees have mostly all registered. We've seen sort of a leveling off of new entrants here. So what I'd like to do is turn it over. Actually, before we turn it over, we'll ask a few questions just to get the group oriented here. We have a pretty diverse number of registrants from a variety of backgrounds. What we thought we would do just to kick things off is ask a few questions to see what your primary areas of interest are. And we'll get those questions up on your screen in just a second here. Here we go. All right, and so your first question, and you'll be able to respond by just clicking one of the radio buttons here, and we're going to leave this question up on your screen for a minute to have people kind of get their responses in, and after about a minute, we'll show the results to everyone, and then we'll move to the second of our three questions in just a second here. Great, this is really helpful. Keep those answers coming in. I think the presenters will use this to help kind of tailor their color commentary as we're going through, and they'll be able to choose, some interesting examples from the work that we've done over the last few years here to highlight some of these concepts. Just a little bit longer, we still have a few more responses coming in. Great, I'm going to close it here. And if you can't see the stats there, we have about 40% of the crowd really interested in microbiome study design. I think that's a super relevant topic. Lots of confounders to be, to be thinking about as we, budget our limited resources and try and, have really strong strategies to uncover these causal links. We have about 20% interested in sampling and stabilization methods, 11% in metagenomic sequencing platforms, and the remainder, about 27%, interested in informatics. So thanks for that. We've got another one that we're gonna put up for the audience here. In terms of where you're getting your primary samples from, and generally speaking, the nature of the research that you're pursuing. So we'll let that run for another minute or so. I think there are new indications cropping up in the literature almost weekly. Someone was describing to me just a little while ago about a fascinating association between the gut, and immune responses that are stimulated by the gut, and also the ocular microbiome and dry eye, which is one of the primary reasons that folks go and visit their ophthalmologist. So a few years ago, if you would have told me about the gut-eye association, I probably would have laughed, but here we are. Great. Thanks for your participation there. I will close it off. And results, not really surprising, but interesting to see and confirm this with data. About 80% of you are interested in the gut microbiome in our sampling stool. A good third are working on saliva and or the oral microbiome, about 20% thinking about skin, 18% looking for environmental inputs for your studies, and then the long tail of weird and wonderful sample types that we all want to analyze coming in the other category. So we'll try and provide some color commentary on the breadth of these as best as we can. Last question before we get into the content. All right. Hang on a second. You have to click. Okay. Because we're covering a range of topics from strategies and study design and wet lab methodologies and analytical strategies, we really wanted to understand your decision-making process for selecting a provider when it comes to outsourcing some or all of these to a trusted partner. So we've got a couple of choices on the screen here about regulatory compliance, breadth of capabilities in the lab, turnaround time, price, and reputation. Great, this is the last of our three questions, and after this one is closed, we'll get into the content, starting off with Evgeny. Just a few more seconds here as we're tallying the votes. Great, I'll close it here. And from our participants, we had the opportunity to select three categories here. And so we might have, a bit more than 100% of a total here, which we do. but in first place is definitely sequencing and analytical capabilities. I think it's important to find a provider that can accommodate a variety of techniques and technologies to suit your study designs. That makes sense. In second place, we have turnaround time. Clearly, decisions on these trials are dependent on data outputs, and those timely outputs are critical. We have price coming in, not surprisingly, reputation, and just a little anecdote. The team that's presenting to you today has a combined total of about 45 Nature, Cell, Science publications on the microbiome. So that should be comforting and very fascinating. And lastly, we have regulatory compliance. But something that we pride ourselves on here is making sure that we have robust quality management underpinning everything that we do. Thanks again for your participation on that, and I'm going to turn it over to Evgeny to kick us off.
All right, thanks a lot, Aaron. Thank you for that great introduction. So I'm just going to grab my pointer here. All right, so I wanted to take you through introduction of the DNA Genotek, how we create microbiome products, but also the different study designs that we undertake in order to validate our products, and some of the considerations that we have discovered that should be relevant to anybody performing microbiome studies. And then after my short talk, CoreBiome's folks, Dan and Daryl, will take over and take you through sequencing. So as you can see here, some of the common considerations from sampling to analysis, as it really starts off with study design, goes through sample collection, and then of course data generation, typically through sequencing, and then analysis of that data. So from DNA Genotek's side, really we take care of the study design and sample collection, in particular through our line of OmniGene products. To those of you that are new to DNA Genotek, we started about 10 years ago, actually more than that, and really started off with saliva collection for human genomic studies. So instead of having a blood draw, you would be able to spit into a tube, and that saliva would be stabilized using our proprietary chemistry, and then DNA could be extracted at the lab. And the beauty of the product is really that these collections can be done at home, and the extraction, that sample can be sent over to any lab, and they can perform the extraction and the data analysis. So we really took that approach and tried it to appropriate to microbiome studies. And we started with our OmniGene gut device. That's the one you can see here in the center, really to allow at-home collection of stool microbiome, again, having a proprietary chemistry in there to stabilize that microbiome and allow that sample to be shipped back to the lab by the donor for analysis. After the success of this product, we expanded our line into OmniGene oral, so oral, really saliva microbiome collection, as well as OmniGene vaginal and others such as gum and plaque. And these products have been used extensively by a number of different researchers and research groups. You can see some of them highlighted down below. So when we talk about microbiome at DNA Genotek, we really talk about stabilizing a snapshot of the microbiome. And by that, we mean the in vivo representation of the taxonomic abundance that's present in the sample. And this can be either bacterial, myco, or even viral abundance. And so there are three different considerations that need to be taken into account when looking at the snapshot. The very first of them is neutrality, and this is really talking about how well does your collected sample that's stabilized in the chemistry compare to a fresh sample, a sample that's less than 3 hours old, provided by the donor, and really has that in vivo biology in it. And the reason that we look at it is that really there's two different main causes that can cause a shift in this neutrality, one of which is incompatibility with extraction. So if your chemistry, stabilization chemistry, for instance, impacts the lytic buffer that's present in commonly used extraction kits, then you will not be able to recover that in vivo microbial profile. And the other one is really contamination, and that's if your stabilization chemistry or physical design of it has significant contamination in it and will mask the microbial profile. Now, this is not as important for gut samples as it is for skin or environmental, where obviously the microbial biomass is much lower, and so that contamination has a much bigger impact. The next one is stability, and this is really capturing how well does that preservation technology stabilize the microbial profile over time. So as I talked about, these samples are typically shipped to the lab, and so during that shipping transition time or even for batch processing, you need to make sure that the chemistry stabilizes that microbial profile. And one main way that the stability can be impacted is if you have biased growth over time. And this is really if the chemistry is providing a new nutrient source or is non-lytic or non-stabilizing. And then the other one is selective degradation of DNA. So when we talk about microbiome samples, we typically think of intact bacteria. However, there's a wealth of DNA available from already lysed bacteria or bacteria in the process of lysis. And so you want to make sure that you're stabilizing that DNA or any other analyte from those sources. And so you want to make sure that there's no degradation occurring. And last but not least, you want to make sure that your device is reproducible. And by that, we mean different extraction aliquots taken from a single collection. They should be representative of each other and of the fresh sample. And one of the causes for that is really if you don't have proper mixing within your device, and your extraction efficiency can also be impacted. So when we're undertaking microbiome validation studies, we look at all of these factors. And really our number one concern, first of all, is having sufficient donor sample number in order to have a rich biodiversity of the microbial species that could be present in the sample, as well as establishing a proper reference. And by that, we mean really a fresh sample. And that's, again, transferred to the lab on cold chain within several hours of producing the sample, and allows us to have a reference that we can compare to and set that as the in vivo benchmark. In addition to that, I won't go through all of these, but really we are testing out different shipping conditions, different ambient temperature holds, even elevated temperature holds in our device. So that's on the left-hand side here, as well as replications of collection and extraction. And we're comparing that not only to the fresh sample, but also to the gold standard, which is really -80 freezing of that fresh sample, holding it for, in this case, 60 days, and then having an extraction from it. And then we would compare our stability over time compared to that -80 frozen sample. And just to give you an example of some of the data that's generated off this, we have here a handlebar plot from a single representative donor, and we're looking at the different conditions that we're testing. So baseline samples, room temperature hold in our OmniGene gut device, as well as elevated temperature. And we're always comparing that, as I mentioned, to the fresh sample. So here we can see that the baseline for the fresh sample compares very well to our baseline, that's the neutrality. And we can see that we're not undergoing any significant changes over our temperature holds, including elevated temperature hold. However, for the fresh sample that was held for 14 days unstabilized, as you can see, and unsurprisingly, we have significant changes of our taxonomic abundances. And we don't just use our own data, we also have several different publications that have used our device. And then, so here I'm just showing one from 2016. Here, multiple adult donors were used for collection into our device, labeled by S, and then doing a freezing, so right away after collection. And in the handlebar plots, you can see that there's a representation of the different donors, and overall their profiles were kept the same across the different replicates within the device, as well as in the freezing conditions. We can also see in the box plots on the right here, in the leftmost one, we can see that the reproducibility within our stabilization was actually better than reproducibility within freezing, and that owns to the homogenization that we do within our device. And we can see also the Bray-Curtis or beta diversity dissimilarity between the T0 and the stabilized samples, as well as T0 and the frozen samples. And you can see that they're very low, which is very good, as it points to low changes between the within the collected device. So moving on a little bit, obviously as part of any microbiome collection, you also need to do extraction to get your analyte of interest out. And typically, this has been done for optimizing for beta diversity or diversity recovery. However, we actually found that there's other factors that are of interest during extraction. So one of these is actually lysis efficiency. So we undertook a Crohn's and colitis collection study and looked at two different taxons in particular, a high abundance and a low abundance taxon. On the left here, you can see for the low abundance taxon, we extracted it using two different methods, a high efficiency and a low efficiency method. And then we repeated that for analysis of the high abundance taxon. And what we found was actually for the effect size generation, the low efficiency extraction method actually required a significantly higher sample size in order to generate that effect size. And this was only for the low efficiency, because with the high efficiency extraction, we're actually able to generate a significant amount of the recovery of the analyte of the DNA for that taxon in order to generate a significant effect size. And again, this is only important for low abundance. So if you're looking at a high abundance taxon, in this case Bacteroides, it actually doesn't matter which extraction method you're using. Both methods were giving us significant recovery of that analyte. And so this is quite important when you have targeted analysis rather than looking at profile wide. In addition to this, we also looked at the recovery of the DNA and the quality of the DNA recovered. So typically when we think of extraction methods, we're looking purely at yields. However, if we're considering long-range sequencing methods such as nanopore sequencing or PacBio, the quality of the DNA recovered can be quite important. And so in this case, we compared 4 different extraction methods. And rather than just looking at yields, we also looked at, by tape station at the quality recovered. And in this case, we can see that certain extraction methods actually give us significantly lower quality of the DNA recovered, and that would impact the performance of that long-range sequencing. And last but not least, as I mentioned, yield is quite important. And based off the statistics that we just ran for the poll, about 40% of you answered that you're looking at skin and environmental samples. And this is where extraction efficiency and yield is actually quite important. So in our study, we looked at infant donors, so down to 8 months of age. And unsurprisingly, we found that the low biomass found in these samples, particularly gut biomass, was actually quite important in the yields that we're getting. And so we compared 2 different extraction methods, the repeat bead beating and the mobile or nucleogen power fecal method. And we found that for the repeat bead beating, we're actually getting significant recovery of our DNA, and it was sufficient for multiple downstream applications such as sequencing. However, for the power fecal kit, this was the non-pro kit, we were actually getting significantly low yields, and in particular for the very young age groups. And this was quite important because for some of these donors, we actually didn't have sufficient DNA for even amplicon-based sequencing, and certainly not for whole genome sequencing. And so when you're considering different sample types such as skin and environmental, again, this is quite important, and it really leads to a requirement for a pilot study to be run in order to verify which extraction method is optimal for your sample of interest. And second to last slide, so I wanted to briefly talk about mock communities and their use. So mock communities, as most of you know, have been more and more used for microbiome studies, and they're really used for quality control. So here I wanted to contrast two different types of mock communities, so biological mocks and DNA mocks. Biological mocks, their makeup is bacterial cells, so these are intact cells that have been purified from pure cultures, and they're typically in the range of 10 to 20 microorganisms that are found in those mocks. Versus DNA mocks can be either exactly the same species as the biological mocks, but simply their DNA rather than the intact cells, or they can actually be synthetic DNA and even in silico DNA for benchmarking databases, for instance, for sequencing. Some of their drawbacks is obviously for biological mocks, as I mentioned, there are only 10 to 20 species typically. So really it's lacking taxonomic complexity. We also don't have any of the sample matrix. So for instance, the gut matrix that's present in a biological sample is not present in the biological mock. And that sample matrix typically carries with it significant PCR inhibitors. And so if you're using a biological mock, you're not actually testing for the extraction efficiency of those inhibitors or the elimination of those inhibitors. You're also not testing for any growth provision by that matrix. And you're certainly not also testing for the diverse nucleases that are found in a matrix, in a sample matrix. And then for the DNA mock, it's fairly similar. So again, we can be lacking the taxonomic diversity for the culture-derived DNA. And we don't have sufficient heterogeneity in PCR errors when we look at in silico DNA. And when we do, we have to generate those and actually put in sufficient care into generating those. So that's quite tricky. However, that's not to say that there aren't any use cases for these. So certainly for biological mocks, the extraction efficiency can be tested with the caveat that these are for select taxons. We can also look at sequencing bias measurement through these biological mocks, and certainly assay reproducibility. Because one of the facets of these mocks is that you're really controlling the input amount. So that's quite important for looking at efficiency and recovery. And then for DNA mocks, again, looking at different reproducibility metrics, such as for your library preparation or sequencing bias identification. So with all that in mind, it's really to say that DNA Genotek has you covered for this. So not only from the perspective of having different devices that are catered to specific sample types and having specific chemistries that are catered for those sample types, but you also get access to our R&D, the know-how that we have generated over the many years that we've been in this. And this comes from study design to data interpretation, and even to scaling through automation for use of these devices. And of course, through CoreBiome, we offer extremely good sequencing and analysis services. And this really provides a rapid, affordable, and highly accurate microbial profiles for any of your microbiome studies. So with that in mind, I'm going to pass it over to to Aaron, our moderator.
Great, thanks again. Just a quick update on housekeeping. On your dashboard, you'll see a tab for questions, and thanks to those who are typing in. I'm trying to keep up with you and answer real time. And for those who have broadly applicable questions, we'll try and broadcast those to everybody. So I've got a couple after Evgeny's presentation here. The first one was about spike-ins. The asker was very interested in absolute quantitation rather than relative abundance. Dan and Daryl, I know you're on the line, and this is something that CoreBiome has put a lot of effort into. Guys, can you chime in on that one?
Yeah, so we actually offer absolute quantification at CoreBiome. The way that we've been doing it to date is by measuring the mass of the sample and then also doing a 16S kind of general qPCR to get a bacterial copy number. Of course, another approach for doing this is using spike-ins, where you spike in a organism or a synthetic standard that you can pull out at a consistent level across all of your samples. That's also an approach that we're working on and developing, but I think both of these approaches give you a way of getting at those absolute abundance quantifications.
Great, thanks, Carl. And the other question that we received was from someone looking to study anaerobic bacteria and if there was any sampling methodology available to accommodate that. And now I can't read your mind about what you were thinking about when you were asking about studying bacteria, but from a sequencing perspective, we can confidently say that if the microbe is present in the sample, its nucleic acids will be protected in the OmniGene devices, and that will allow for a bias-free, approach to studying these things from a sequencing perspective. But from the point of view of culturing these anaerobes, I have to ask for your patience and stay tuned. This is something that DNA Genotek and CoreBiome are working on, and hopefully in the next webinar we'll have some interesting updates for you there. Okay, with that, thanks for the questions. Keep typing those in. We're going to switch to the next presenter. Daryl, you are up. Take it away.
Great. So I'm going to tell you today about work that we've been doing at CoreBiome to improve the accuracy, the resolution, and the reproducibility of microbiome data generation. And so I'll be talking about this next phase in the microbiome analysis pipeline, data generation phase. So at CoreBiome, we offer a variety of microbiome profiling services. This includes both 16S and shotgun approaches. We offer both deep shotgun and then also this approach that we call booster shot, which is a shallow shotgun sequencing approach that we've pioneered that allows you to get high resolution microbiome data in a cost-effective manner. So we a couple of years ago announced the first metagenome for under $100. We also have a service called StrainView that allows you to do whole genome sequencing and assembly on collections of isolates like strain banks. And then finally, we offer custom analysis services called Core Plus analysis. And in this service, we really act as a discovery partner. So we help you derive insights from the microbiome and discover biomarkers and make other discoveries. So the genesis of the booster shot approach, or our shallow shotgun approach, was really this recognition that there's this information gap in microbiome data generation. So on one hand, you have 16S sequencing, which has the benefit of being really cost effective and allowing you to do very large scale studies, which are often necessary to detect a signal in the dynamic microbiome. The disadvantage of 16S sequencing is that the resolution is relatively low, so often you can only get genus or family level resolution, so you're not getting to the fundamental unit of processing within the microbiome, which is a species or a strain. On the other end of the spectrum, you have deep shotgun sequencing, where it's very information rich. You're able to detect species and strain level resolution. You're able to observe the functional content of the microbiome, so the genes that are present, and you're able to assemble novel strains and genes out of that deep shotgun data. But it has the disadvantage of being very costly and not practical for running very large scale microbiome studies with lots of samples. So we really recognized that there was this gap here where we needed methods that were both high resolution and cost effective that could be run on large sample sets. And so booster shot was our solution to this. And with booster shot sequencing, we're able to get species level profiles. So, really comparable to the type of data that you get with shotgun sequencing. And also really highly accurate profiles of the enzymes, genes, and metabolic pathways present within the samples. So we described this approach in a paper last year that was published in M Systems. And I'm going to show you a little bit of the data from that paper. So this is a data set or analysis of the human microbiome project data for different body sites. And when you subsample down to different read depths, what you can see is that at around 100,000 reads or so, you're able to recover the full patterns of alpha diversity within this data set. Likewise, at a depth of a couple 100,000 reads, you're also able to recover patterns of beta diversity. So to be able to distinguish these different body sets from one another using the Human Microbiome Project data. Finally, this shallow shotgun sequencing approach also has really comparable power to deep shotgun sequencing for biomarker discovery. So this is an example of some published data from a study of type 2 diabetes. And what's plotted here are the p-values of biomarkers that were picked out that were associated with the diabetes phenotype. And what you can see is that when you use very deep sequencing compared with much shallower sequencing, a couple orders of magnitude less sequencing data, you're able to pull out really comparable p-values and all of the same biomarkers that you would get with the very deep data. So what this allows then is the ability to really scale up and do large-scale studies and get much higher resolution than you would get using 16S sequencing. So we've used Booster Shot to analyze samples collected in all of the different DNA Genotek collection devices that Evgeny mentioned in his talk. And what you can see here is that the beta diversity patterns that we observe for the stool and oral and vaginal samples that are collected in the DNA Genotek kits really recapitulates the patterns that were seen in the Human Microbiome Project. So this is a recent publication of some of the Human Microbiome Project data based on shotgun sequencing. And we see very similar patterns of beta diversity. So with CoreBiome, every project that we run includes an interactive analysis report. You can go to this URL here and see an example of these reports. And it includes both some interactive data visualizations, as well as a quality dashboard where you can see the read depths and sequencing quality and a lot of the quality metrics associated with the samples. And then also allows you to download both the raw data and then a lot of the different data products that are generated in the course of this analysis. So OTU tables, gene and enzyme tables, all the different analysis products that you need to kind of do further analysis. So we've also been working on really testing and improving the reproducibility of the data generation at CoreBiome. I'm going to show you an example of a study that we've run where we collected samples from 20 subjects using the DNA Genotek OmniGene gut kits. And then we ran a bunch of different studies on these. So first we tested intra-run variability. So we took 3 replicates. They were processed on the same plate, by the same technician, taken all the way through to sequencing. And then we also ran another study on inter-run variability where we took three sets of replicates of these samples and processed them on 3 different plates and took them through on 3 different sequencing runs. So all the way from extraction to sequencing. And then we also looked at inter-operator variability. So this is 2 different technicians in the lab each processing a replicate set of these samples, on a different plate and taking them through to sequencing. So this is an example of some of that data. So this is just 2 of those 20 subjects, but what you can see is that both the intra-run and inter-run variability, as well as the inter-operator variability, are all very low. So we get very, very consistent profiles for this assay across all of these replicates. And this is just a quantification of the full data set where we're looking at the coefficient of variation between the Shannon diversity measurements of these samples And what you can see is that for all the technical variables, so interrun, and interoperator, we're seeing very, very low CVs for Shannon diversity that are really dwarfed by the actual biological variation or the between subject variation. So I've told you a little bit about some of the approaches that we're taking at CoreBiome to generate really high resolution and highly reproducible data. And now I'm going to turn it over to Dan, who's going to tell you about some of the things that you can do with this data downstream.
Thanks, Daryl. Let's see here. So that's a little bit more on the data generation side. And I'll talk about some of the implications of different types of data on how we would do this, how we would analyze these data and design studies for these types of data. So, I think what Daryl talked about earlier is a solution to like the first major problem that we've been trying to solve, which is helping people get big data. And that includes, both putting into place more affordable methods so that you can actually do shotgun sequencing on whole studies, and also making them fast. And actually, we were looking back at our stats at CoreBiome, and CoreBiome to date has sequenced and analyzed over 50,000 shotgun samples, and that's actually just in the last 2 years. And this is actually just about double the number of publicly available metagenomes in the EBI magnified database. So this is really, just putting shotgun sequencing into the realm where you can actually do it instead of 16S sequencing on entire studies. We have groups doing thousands of samples in a single study that are all shotgun sequencing. The other thing you have to do is know how to use the big data once you get it. And so I'll talk a little bit about that. I also noticed, I have an academic lab, and I noticed in my lab that there were a lot of groups who they had ways of getting data, but they didn't always have people in their research group or in their lab who knew all the ins and outs of microbiome study design, analysis, and so on. And so one of the things that we've tried to do at CoreBiome is encapsulate all of the knowledge about best practices for how should you design a study based on what you're trying to test. How do you deal with various confounders? What are the main confounders, and how should you design the study to either account for them by the design itself or through the statistics? What are the best statistics to use given the different types of study design? Longitudinal data, for example, has some very different types of statistics that are available that can be very powerful. Also integrating different sources of data, like multi-omics data, we would call it. So, we have some studies integrating host genotype or host gene expression with the microbiome, and integrating the microbiome with metabolomics data. And so, those are some of the capabilities that we have at CoreBiome that we can basically help people get access to, for doing their science. There's also a growing need for machine learning in the field, especially as we see multiple different types of clinical outcomes where you can actually stratify patients ahead of time using the microbiome by their clinical response. So you can tell responders, non-responders, for example, in some immuno-oncology drugs, using the microbiome that they walk into the hospital with. You can also do this for certain types of infections. So that's something that is very important for patient stratification. And then, of course, there's data visualization. So finding ways of actually making the findings interpretable. And all of these things we've put into place in what we call Core Plus analysis. So there's kind of the data factory arm of the company that's highly reproducible. We can do massive shotgun sequencing studies. And then we have all of the custom analytics and consulting under the Core Plus analysis service. And so now I'm going to show just one example of some of the types of analyses that that we can do with these new data generation capabilities. So this is a study that was recently published by my academic lab that CoreBiome helped out on, and was also sponsored by General Mills. And this in this study, what we did was to collect daily shotgun sequencing for 2.5 weeks from 34 people. And each one of these boxes represents the time series from a single person. So you can see how each person has kind of their own profile or fingerprint, and some people are more variable than others. So this, for example, this is Abby Johnson's microbiome. She was the postdoc in my lab who led the study, and she had one of the most stable microbiomes, and I think she was proud of that. She gave me permission to show her data here. So so we can start to actually see the species level variation because it's shotgun sequencing. And it's the first time that we've seen daily variation at the species level in a cohort of subjects. And this was done using shallow shotgun sequencing. This was actually ultra shallow, and we were able to do the whole study for the cost of getting just one conventional deep sequencing time point from each person. And yet we get all of this additional longitudinal information. And then one of the types of things that we would want to do, obviously, is incorporate exposure data or other omics data. And in this case, this is the food ohm. These are all the foods people were eating, and those were collected every single day, so we can pair those up with the microbiome. And immediately you can see the food data is also highly variable and somewhat personalized. A couple of people had a very interesting diet. These 2 people were consuming Soylent for the entire study, which Abby said is apparently what happens when you have computer scientists in your study. So they provided an interesting little sub-analysis. But the richness of the data allowed us to do things like analyze the stability of the microbiome, which is something you really wouldn't be able to measure accurately without multiple samples. So this is the species level stability of the microbiome. Basically, how much does it change from day to day in a person? And we found that this was highly correlated with the diversity of a person's diet on average. So if someone tends to choose a diverse set of foods, then they tend to have a more stable microbiome. Of course, this is just a correlation. We don't know that it's causal, but it was very interesting and something that we'd like to follow up on. And again, because of this dense longitudinal data, we can actually find out what people's own personalized diet microbe associations are. So we can look at how one food varies from day to day in a person and how a particular microbe varies from day to day in that same person. And we see that some of these are very conserved. So for example, cakes, cookies, bars were positively associated with Roseburia inulinivorans in multiple subjects. And inulin is actually a common food additive, bulking agent, or fiber adding agent in a lot of fiber bars and things like that. So this kind of makes sense because inulinivorans means eats inulin. But there were other foods such as dark green vegetables or pork where we saw that they were positively associated with a particular microbe in some people and negatively associated with that microbe in other people. And this might be one explanation for why it's sometimes been hard for the field to find consistent food microbiome associations in a large cohort, because most studies haven't had that dense longitudinal data, so they haven't been able to do this type of personalized association within each subject. So you can read more about that article following the link there. And if you're interested in adding diet to your study, incorporating other omics data, adding machine learning, help with study design, any of that stuff. As I said, we've kind of tried to make that accessible to anyone who needs it, academics, biotech, pharma companies, through CoreBiome. And lastly, Daryl also mentioned that we have deep sequencing. So what we've done here is we've used the same technologies that allowed us to make shallow shotgun sequencing affordable, and we're applying many of those to make deep sequencing more affordable and scalable. So this is something that a number of our customers at CoreBiome are continuing to do, is when they run a study, they'll start off the study with shallow shotgun sequencing using booster shot, which is 2 or 3 million reads usually, and then and that might be thousands of samples, and then they'll zoom in on a subset of samples for deep sequencing, and this is basically the same thing people have been doing for years, except you're skipping 16S sequencing entirely. So you just go straight to shallow shotgun sequencing, and then you follow up with deep sequencing on a subset of samples that you think are most interesting. And this, if you want to read more about some of the interesting types of features we can get out of the deep sequencing, there's a link to a paper that my academic lab published last year that identifies some enzymes associated with U.S. immigration. So, I think we'd like to stop here and save a little bit of time for any questions that you might have. I know there was some interest in study design. A lot of people expressed that. So, so maybe we can talk about some more subtleties there. But in general, these are the main steps, and these are some of the solutions that we've tried to put in place in an industrial setting to basically kind of grease the wheels of science and help more people do better science faster. And with that, I think we'll close and take any questions.
Great, thanks so much, Dan. And thanks to the participants for the steady flow of questions. Really impressive and thoughtful. I'm going to take a few minutes just to kind of recap some of the highlights, but the question app is still open. So as we're going through these, keep them coming. If we don't get to you during the next, 5 or so minutes, we'll follow up separately via email. There have been quite a few requests for a list of references and so forth. We'll provide that through the Microbiome Times, just so you have a tidy reference list of the publications that we referred to here. Dan, I think the first one's going to go to you. The question is around power calculation. This came up a few times, but the generalized question is, I have this interesting phenotype and I've got microbiome data from my participants. How should I be really thinking about power calculation to make sure my study is robust?
Mm-hmm. That's a great question, and one that we've been increasingly able to answer the more published microbiome studies we have. And my favorite way to address that personally is really to find what I think are comparable outcomes of interest that have already been studied, and preferably ones that have been studied, a few times. And for example, you could say, take diabetes. There are quite a few diabetes studies focused on the microbiome that have shotgun sequencing, and you can use those studies to understand, with a given sample size and controlling for certain confounders, what were those researchers able to detect. And this is a nice way to deal with the challenge that the microbiome field is moving so quickly that for many outcomes, exposures, diseases, we don't know exactly what the study size will be. And so, sometimes, like if I'm writing a grant, I will say, well, we think that the study size will be roughly comparable to the effect seen in obesity or diabetes. And then I'll show what the performance calculation is based on those existing studies, and then map that over to the study that I'm designing or proposing.
Great, thank you. Next one is for Evgeny. There have been quite a few questions about metabolomics and if we have anything in the OmniGene line that can handle that.
Mm-hmm. For sure. So while the OMR 200 product is not designed for metabolomics, the chemistry is specific for DNA from microbiome. We are developing solutions for metabolomics from gut. And so we actually have at a prototype stage a product that we can start providing to select customers and select testers based on your needs. So we certainly please follow up with us directly, and then we can provide you first of all data for it to support our product, but also any test kits that can be made available.
Yep. Yeah, and to do that, just send an email to info@dnagenetek.com. Reference that you are on the webinar today, and we'll be happy to provide whatever information you need, whether that be prototype kits or what have you. So we're looking forward to that. Next one goes back to CoreBiome. The question is, and this came up again multiple times, folks want to know about the breadth of applicability of booster shot and shallow sequencing, especially, in the context where certain microbial environments might not be as well characterized as stool. What are your recommendations there?
Sure. Generally, we would use booster shot for host-associated microbiomes, and we've done any range of hosts, poultry, livestock, a number of mouse studies, and plenty of human, because those tend to be better represented in the reference databases. If you're looking to do environments that are more novel, like ocean or soil samples, or some host that's very very novel, like a frog or something like that, then you probably would either want to do deep shotgun sequencing so that you don't have to rely on the reference as much, so you can assemble and bin putative microbes from the shotgun data, or we would use benchmark, which is our 16S method, and that has some tricks on the molecular side in it that make it a much more broad range than some of the conventional 16S methods or amplicon methods taxonomically. So that would be my recommendation: is either do 16S sequencing for bacteria in those environments, or go straight to deep shotgun sequencing.
Great, thanks, Dan. There were another batch of questions oriented around some of the modules or data sources that CoreBiome pulls from, in particular databases and algorithms. Would it be possible to provide a general overview? Sure.
CoreBiome is using data, genome data that are curated from RefSeq, and we have that annotated with taxonomy as well as functions, and we can map those functions to various ontologies. So KEGG, eggnog, COG, UniProt, and others, and other ontologies. So we're really leveraging what's available in these highly curated, carefully designed databases, and then doing some additional kind of organization and cross-mapping between them, so that we have different types of features that people may be needing or used to interpreting. And those are all standard. We also have the ability to incorporate other types of databases, and we've done some projects looking at carbohydrate degrading enzymes from the Kzyme database using deep shotgun sequencing. So there's quite a bit that's there already that's pretty readily available. And then in terms of custom databases, the sky's the limit.
Great. And last question, and apologies to those that we didn't address during the seminar. We'll follow up with you separately. But the last question is, I'll say a generalized look forward on the CoreBiome roadmap, specifically what's happening with RNA, sort of the regulatory accreditations, roadmap, and so forth. Dan and Daryl, could you provide a quick look ahead for CoreBio?
Yeah.
Daryl, you want to do that?
Yeah. So you know, the big focus of our R&D recently has been on developing metatranscriptomics assays. So that's really the next thing that will be coming down the pipe at CoreBiome. So, you know, if you're contemplating doing those sorts of, like, you know, gene expression metatranscriptomics measurements, certainly get in touch with us, and we'd be happy to, you know, tell you where we're at and hear about your projects. And then the other thing that we've been working on putting in place recently is CLIA certification for customers that might need, you know, more like sort of medical, clinical grade sequencing. So also look for announcements on that front soon.
Fantastic. Thanks to the presenters, and thanks again to Microbiome Times for coordinating. And thanks to all the participants for your active engagement. I was really impressed with the questions, and we'll follow up separately with anyone that we couldn't get to during the allotted time. Thanks again from DNA Genotek and CoreBiome, and we look forward to working with you soon.